This is the second video, and in this video we will discuss about the types of chemokine and how the chemokines are classified. Chemokines are grouped and named according to their amino acid composition. Chemokine may also be grouped according to their function, such as whether they are inflammatory or homeostatic. Inflammatory cytokines are produced when inflamed tissue releases cytokine such as TNF, that means tumor necrosis factor. Cytokines are small in size, which is 8 to 10 kilodalton, and it contains around 70 to 125 amino acid residues. Members of chemokine family can be divided into four main categories depending upon how the first two cysteine residues are placed. According to this, chemokines are divided into four groups. First is CC chemokine, second is CXC chemokine, third is CX3C chemokine, and the fourth and last is XC chemokine, which is also known as C chemokine. Now I will discuss briefly about the CC chemokine. These have two adjacent cysteine near the N terminus. These are also called as CC chemokine ligand, which is CCl. There are 27 members of this group have been identified and are numbered CCl1 to CCl28. This point is little bit confusing because 27 members but the number is 28. This is so because CCl9 and the CCl10 are almost similar. So if we count by number, then there are 28 CCl, but if we count according to function or by function, they are only 27 because CCl9 and CCl10 are similar in function and appearance also. Most of the chemokine contain 4 cysteine, which is C4CC, but some of them contain 6 cysteine, which is C6CC. CC chemokine trigger the movement of various immune cells like monocyte, natural killer cells, and dendritic cells. The second type is CXC chemokine. These chemokine are similar to CC chemokine because CC chemokine have two cysteine residues and the CXC chemokine also have two cysteine at N terminus. But the main difference is CC chemokine is not separated by any amino acid, but CXC chemokine are separated by an amino acid, which is denoted by X. There are 17 CXC chemokine have been identified in mammals, and they are divided into two main groups. The CXC chemokine containing the ELR amino acid motif tends to be angiogenic, where ELR negative CXC chemokine are mainly angiostatic, with CXCL12 being a possible exception. ELR amino acid motif is a special type of amino acid motif. The full form of ELR, the ELR is the short form of amino acid. Denotes glutamic acid, L denotes leucine, and R denotes arginine. And ELR positive and ELR negative are the two terms which we describe later. With a specific amino acid motif, ELR immediately before the first cysteine are termed ELR positive, and those without such a motif are described as ELR negative. ELR positive CXC chemokine interact with chemokine receptor, which is CXCR1 and CXCR2, on the surface of neutrophils to induce their recruitment, and the ELR negative CXC chemokine mainly act by attracting lymphocytes towards them. Here there are two terms, that is angiogenic and angiostatic. They are important term. Angiogenic means formation of new blood vessels from pre-existing vessels, and angiostatic is just oppose the angiogenesis, which means inhibit angiogenesis. Angiostatic inhibit angiogenesis, and angiogenesis means formation of new blood vessels from pre-existing vessels. The third group of chemokine is XC chemokine, which is also known as C chemokines. This is the member of third group of chemokine and are different to all other chemokine because they have only two cysteine, one at the end terminus and other at downstream. Only two of these chemokines have been identified yet, and these are called XCL1 and XCL2. The fourth and last group is CX3C chemokine. These members of this group have two cysteine at the end terminus like the CXC chemokine and CC chemokine, but they are separated by three amino acid which is denoted as X3.